Hi, Michelle Glass here, and welcome back to another installment in our Chapter 19, Lecture 19 series. And our topic for this video are the different types of white blood cells. Now, we can actually group these cells based on their role in our immunity. So as we start moving into the lymphatic system in Chapter 22, we'll see that we can kind of break our defenses into two categories, one being nonspecific defenses and the other being specific defenses. The key difference here is that your nonspecific defenses, they don't know who that pathogen is. They don't know what it's doing there. They don't know any of the details. They just know that that is a foreign cell that doesn't belong and needs to be removed by the body. In the case of specific defenses, this is where you know exactly the who, the what, the where. In this case, these cells are recognizing specific pathogens to the strain and then attacking accordingly. This is with specific defenses, this is how we can have that memory to specific pathogens that we've been exposed to. So we might get sick the first time, but the second time and all subsequent times, we are not going to have any particular problems. When we talk about nonspecific defenses, which are going to be part of our Unit 2, we are going to see that there are 4 types of white blood cells involved here. We have the Neutrophils, Astana Fills, Basophils, and Monocytes. Now, all 4 of these have been named based on how they stain and what they look like under the microscope. You have a whole discussion of that in your chapter. So if you are interested, or if you think you might need to know that in your future programs, like if you're going to be a medical lab technician, then go ahead and start doing that research. But for us in this course, we really just need to describe what they're doing in the body, not so much what they're looking like in the microscope. On the other side, we have our specific defenses. So these are our lymphocyte cells. We're going to talk about these in more detail when we get into Chapter 22. So we'll talk about them a little bit as part of this unit. We will introduce them as the T cells, which you've probably heard of before, B cells, and then what are called NK cells. The NK there stands for natural killer. And then we'll talk about these again when we're investigating specific defenses in our Unit 3. So we are going to spread our Chapter 22 information out across 2 different units. Okay, so our emphasis will be here on our non-specific defense cells. So let's talk about each one of these. So let us begin with that neutrophil. The neutrophil is the most numerous in circulation, so you can use the first sound there as your memory device. Neutrophils are numerous in circulation. These are the cells that are going to be the very first to show up at a site of injury, and their job then is going to be to eat up or phagocytize any pathogens that have already been marked by antibodies. So again, this is a case where they don't know who that pathogen is. They just see that it's been flagged by having antibodies attached. You can think about these antibodies as like the salt and pepper that makes those pathogens tastier. So this is going to be one of the ways that those neutrophils are going to recognize and then engulf these pathogenic cells. We also see that neutrophils are going to release 2 important types of chemicals. Prostaglandins, which are eicosanoids. These are local hormones that are released. So this is a paracrine form of communication. They're going to increase the capillary permeability, creating local inflammation. So this is going to bring more blood to this particular area, but then also sort of close this area off, making it more difficult for this pathogen to spread to other parts of the body. And we will also see the release here of leukotriene. This is a hormone that will attract other phagocytes. So this then is an example of a positive feedback. The neutrophils come in, they release leukotriene, which is going to bring other neutrophils and other phagocytes to this area so that we have a quicker immune response here. And remember, our white blood cells have positive chemotaxis, so they're going to move toward this particular chemical. Our next category are the Astana Fills. Eosinophils are also attacking antibody-coated objects, but in this case, we have the word object because we're getting both tiny things like bacteria and protozoans, which are small but quite a bit bigger than those bacteria. And then also we can see cellular debris is being removed here, and big stuff like flukes and roundworms, things that are visible to the naked eye. Because like those flukes and those roundworms and even some of those protozoans might be too big for the eosinophil to engulf, we see that the eosinophils will release our chemical surrounding these cells. So we're going to get the release of both nitric oxide, which is abbreviated NO, and also cytotoxic enzymes. So cytotoxic enzymes, we're going to start breaking down these flukes and the roundworms from the outside. So because eosinophils are able to target these big parasites, we will see eosinophil population increased if you have a parasitic infection. So this would be one way to identify a parasitic type of infection from something else. We will also see that eosinophils are increased when you're having an allergy response. So we will see them also responding to allergens. So I like to use my E sound to remember eosinophils are targeting parasites and allergy. So you can think of parasites as being icky. So I know that's not an E, but it sounds similar. Or eek, it's a parasite. So that's an E sound, right? And then for allergy, you can be like, achoo, to help you remember that one. Okay, moving on. Last little bit here is that your eosinophils will actually reduce inflammation. So the neutrophils were triggering inflammation with prostaglandins. We also have another group of cells called mast cells. Mast cells are discussed in chapter 4. These are cells that are found in our connective tissues. These cells release histamine and heparin, both of which are chemicals that trigger local inflammation. So the neutrophil and the mast cell here are triggering inflammation. Eosinophils are reversing that, so they're reducing that. So that then would be an example of a negative feedback. Neutrophils, mast cells trigger inflammation, and then the eosinophil comes along and reduces that. Okay, now we're ready for our next one, which is the basophil. Basophils will accumulate in damaged tissue, and they will be responsible also for local inflammation. So just like mast cells, basophils release histamine and heparin. The histamine is responsible for dilating the blood vessel, and then heparin is preventing the blood clotting. So this is what is triggering that local inflammation to occur here. So basophils work together with mast cells to trigger local inflammation, but they are two separate types of cells, but we see them working together quite a lot. Well, and then here I have complete typo, so let's just, well, can I mark through it? No, oops, okay. Attract is the word we want here, so excuse that. The basophils are going to attract eosinophils and more basophils. So we're going to bring another kind of positive feedback mechanism here to this area. And then our final population to talk about are those monocytes. Monocytes are going to be in circulation for about 24 hours and then leave. And when they leave, then they're going to be called macrophages. So monocytes give rise to macrophages. Macrophages, we're going to see lots and lots of places in the body. We've already talked about them before. We actually talked about them in AMP1. We talked about them earlier in this chapter when we talked about the removal of those old and damaged red blood cells. The macrophage is a very important, very aggressive phagocyte. We're going to see it located outside of circulation. So we've already seen them in the spleen. We'll see them in other lymphatic tissues. We'll see them in connective tissues. So we'll see them around the body. They will attract neutrophils, monocytes, and other phagocytes. so that if they are in a particular location from injury or infection, they're going to help to bring these other white blood cells there, and also fibroblasts. Fibroblasts will be important in helping to build scar tissue if we have a damaged area. So it's going to help to lay down those collagen fibers. All right, so that's it for now. Stay tuned.